So, last but not least, I take a lot of pleasure to introduce the next speaker, Professor of Molecular Pharmacology, Niels Voigt. Now, Niels is actually my supervisor from my master's and PhD project that just finished a couple of months ago. So it's a particular pleasure to welcome you. Niels completed his MD in Dresden University and then completed postdoctoral studies across multiple prestigious universities across Germany, including Mannheim, Heidelberg, and finally Duisburg-Essen. And now he is a professor at Göttingen University. He has a specialization involving ion channels and calcium homeostasis in the healthy and diseased heart, with a particular focus on the molecular mechanisms of atrial fibrillation. So Niels, thank you very much for coming today, and we look forward to your talk. The floor is now yours.
Yes, thank you very much, Will, for this warm-hearted introduction. Thank you very much. And I think it's a really great pleasure to be here. Good afternoon, everyone. Thanks also for Nanion for the invitation to be here. Thanks, Niels. Thanks, Ali, for organizing that. Also, thank you for placing me at the end of this meeting, because all the techniques which I will present to you have already been introduced to you by Bjorn and Bettina, the previous speaker. So, thank you very much. As Will already introduced, during the last, let's say, 15 years, I've been working on the molecular mechanisms of atrial fibrillation. It's the most frequent cardiac arrhythmia. And by doing that, I had the honor to contribute at least a little bit to this great increase in knowledge on atrial fibrillation, on molecular mechanisms on atrial fibrillation, which is plotted here. I did a few years ago, I did a PubMed search on this, and you can see that there's a huge increase in knowledge. However, what puzzled me a bit is the fact that despite this huge increase, the amount of antiarrhythmic drugs that were approved during the last 20 years is almost neglectable. And what we felt is that potential reason for that is most of these studies were performed in animal models, where it is well known that the the cellular electrophysiology is different from the model and what we see in patients with atrial fibrillation. So therefore, we were wondering whether the iPS technology introduced by the previous speaker and also by Björn could help to overcome this bottleneck, this translational bottleneck, and develop new models of atrial fibrillation. So several groups, also including the one from Lukas Czüganek in Göttingen, our colleagues, developed methods to differentiate iPS cells specifically into atrial cardiomyocytes using retinoic acid. So, and you can see here, these cells nicely express atrial-specific proteins, like here the myosin light chain for the atrial and ventricular forms. But we were also interested whether they express an atrial phenotype as well. So in the first step, actually, that was one of the first experiments Will did in our lab as a master student. You can imagine this quite a few years ago. But we used fluorovolts, also introduced by Björn, to measure action potentials in these cells. These were stimulated at 1 Hertz. They were not spontaneously beating, and we measured action potentials, and you can clearly see that the atrial action potential is shorter compared to the ventricular one. So now, as a next step for the full electrophysiological characterization, we got interested in the automated patch clamp systems. And first measurements were done here in collaboration with Nanion in Munich. And so Will came here and with the cells and did experiments. And later we were lucky to get the funding to buy our own SyncroPatch system. And in contrast to the manual, very labor-intensive patch clamp technique, the system pipettes automatically the cells into the patch clamp chip, and this chip enables us to measure 384 cells at the maximum in parallel. So this is a huge increase in throughput, and by this we could measure the ventricular and the atrial cells at the same day of differentiation and all in parallel. And The first thing what we measured is we measure the L-type calcium current, because this is what we well known, where we know the protocols very well. And we used first a depolarizing ramp pulse from -80 to -40 millivolts to inactivate fast sodium currents. And the further step to +10 millivolts activates a typical L-type calcium current. And in accordance with the shorter action potentials that we observed in the atrial cardiomyocytes, the L-type calcium current was smaller in atrial cells compared to the ventricular ones. As a next step, we were interested in atrial-specific ion channels, such as the acetylcholine-activated potassium channel, ICa-ACH. To measure this channel, we applied a ramp protocol from -100 to +40 millivolts. Please note again, this channel is only expressed in atrial cardiomyocytes, not in ventricular ones. So in the absence of M-receptor agonists, of any stimulators, this channel is closed, and only the inward-rectifier potassium current IK1 is active. And we can see this current here as a in response to this ramp pulse, as a typical inward rectifier current with high conductance as a negative potential and the low conductance at the positive potentials. We found IK1 currents in both ventricular and atrial iPS cardiomyocytes. If we now apply acetylcholine, or the more stable analog carbachol, we observed a dramatic current increase in response to carbachol, which is defined as the ICa-ACH channel, which we observe in atrial, but not in ventricular iPS cardiomyocytes. So now, using this specific ion channel and the high throughput method of the SyncroPatch system, we use to characterize functionally our cellular preparations. And as you can see here, in the ventricular preparation, only 1 out of 54 cells show the response to carbachol, whereas in the atrial preparation, 85% were responsive to carbachol. And this is, to our experiment, similar to what we observe also in cells which we isolate from patient biopsies. So taken together, the first part of my talk, we conclude that retinoic acid enhances the differentiation of iPS into cardiomyocytes with a clear electrophysiological profile. So the next step, to answer the question whether we can use these cells as a model for atrial fibrillation, let's take a look on the most important paradigm in atrial fibrillation. Atrial fibrillation is a self-progressive disease, starting from, at the beginning, from silent or paroxysmal episodes, which later develop in long-standing persistent or even permanent atrial fibrillation. So this self-promoting characteristic of atrial fibrillation is supposed to be due to structural and also electrophysiological alterations that are summarized as so-called remodeling. And Maurits Alessi in 1995 summarized this nicely as AF begets AF. And since then, many animal models have shown that the high atrial frequency itself is sufficient to induce this atrial remodeling and increase the susceptibility of AF. And even more, studies have shown that we can use isolated atrial myocytes, apply the ventricular high frequency, and induce tachycardia-induced electrical remodeling. So we were wondering whether we can induce electrical remodeling by applying tachycardia to iPS-derived atrial cardiomyocytes. So we stimulated these cells electrically at 3 Hertz for 24 hours and compared them as controls with cells paced at 1 Hertz. And the results we compared to our previous results obtained in native human atrial cardiomyocytes. So this is not an iPS-derived cardiomyocytes. This is a cell which we isolated from a patient biopsy. So first we measured again the action potentials, and similar to what is observed in atrial cardiomyocytes from patients, we observed a shortening of the action potential duration after 24-hour tachypacing. So the action potential shortening and the hyperpolarization of the resting membrane potential are both hallmarks of the electrical remodeling in atrial fibrillation. So it is well known that these changes in the action potential are due to alterations in the ion channel activities. So, for example, the reduced L-type calcium current contributes to the shortening of the action potential, as well as the increased ICa current. The increased ICa also contributes to the hyperpolarization of the resting membrane potential in patients with AF. So we are now interested in whether we can see these phenomena also in iPS cells paced at 3 Hz. And indeed, using the SyncroPatch, we observed that after 3 Hz tachypacing, we saw a clear reduction of the L-type calcium current, which is in correspondence with the what we observed in native cells. However, the ICa current was unchanged after 24-hour electrical pacing. So we conclude that 3 Hz 24-hour tachypacing partially induces major hallmarks of atrial fibrillation associated electrophysiological remodeling in atrial iPS cardiomyocytes. So, but we were still puzzled because I did my MD thesis many years ago in the lab of Dobromir Dobrev by measuring a lot of ICa currents. So we were wondering why we do not see an increase in ICa. So maybe there's a whole tissue construct required for ICa remodeling. So we took advantage of the expertise in our institute especially from Wolfram Zimmermann to do engineered human myocardium. So we use fibroblasts and collagen and cast these little rings, and after around 30 days they start beating, and we generated so-called atrial engineered myocardium, and again we paced them for 24 hours at 1 and 3 Hz. We used the sharp electrode technique to measure action potentials in these little construct, and again we observed an action potential shortening as a hallmark for AF remodeling, but we did not observe a change in the resting membrane potential. And as I've shown you on 2 slides before, ICa is the major determinant of the resting membrane potential. So we assume that resting membrane potential, the ICa is also unchanged in these tissue constructs. So next we hypothesized that maybe we need a longer pacing duration of these EHMs. So we paced them for 24 hours for 7 days at 3 Hz electrically, but unfortunately, none of these EHMs survived the pacing. And there are different hypotheses, but one could be that just the redox or the oxidation or the energy you apply by crude electrical pacing to these constructs will just kill these engineered heart tissue. So next, we took again advantage of the expertise in our institute because the group of Wolfram Zimmermann generated an iPS cell line which expresses crimson, an optogenetic ion channel. So this has been already introduced by Björn Nolman, and it allows us to stimulate these engineered heart tissues with red light pulses. So we developed not so sophisticated like the ones here in-house from Nanion, but we developed very working LED-based stimulation plates that allows us to apply short light pulses at a certain frequency. So again, we paced these EHMs optically for 24 hours at 3 and 1 Hz, and similar to the electrical pacing, we saw a clear action potential duration shortening, but 24 hours were not sufficient to change the resting membrane potential. But if we now pace at 7 days, which these EHMs survived perfectly fine, we saw, in addition to the shortening of the action potential, a clear hyperpolarization of the resting membrane potential, which leads us to the conclusion that there may be an increase in ICa also in these cells. So to test that was actually one of the reviewers' questions, and Will worked very hard to answer that. So we paced the cells in culture and measured IK1 and in these after 24-hour pacing, and as expected, after 24-hour pacing, we did not observe any change in the IK1 density as shown before. But if we now pace these cells for 7 days, we see a clear increase in the IK1 density. So, in conclusion, the hyperpolarization of the resting membrane potential and the upregulation of the IK1 occur after 7 days of tachypacing. They may therefore represent markers of AF progression in vivo. So, and to summarize my talk, I took advantage of the latest AI developments. Again, not as much sophisticated as Björn Nollmann. So I asked ChatGPT, are iPS cardiomyocytes superior to animal models for studying AF? So, and I probably couldn't word a more diplomatic answer as the system did. So both have their strengths and limitations and for studying atrial fibrillation, and it is difficult to compare their superiority. Animal models provide a more comprehensive approach to studying AF, while iPS offer the advantage of studying human cardiomyocytes and patient-specific factors. The choice of the model depends on the research question and the feasibility of the study. And with this, I, of course, would like to thank all the contributors to this work. First of all, Will Seiberts, who worked very hard during the revision and doing all these iPS cell-based, the cell-based experiments. Tony Rubio did all the sharp electrode experiments. I would like to thank the support from Wolfram Zimmermann and his group. I already mentioned them. Lukas Czyganek for the iPS cell, for providing the iPS cells, and many other collaborators, and in particular Nanion for their great support during the last years. And I think that Will is a perfect example of our great collaboration. Thank you very much, and thank you very much for your attention.
Thank you very much, Niels. Wonderful to see the data presented in such a nice way. Thank you. Are there any questions from the audience at the moment? Ellie, maybe anything online for us? Or maybe people want to get to the beard. One question from David here. I'll give you the microphone, Dave.
A simple question on the animal question. Which types of animal are they all useless?
Pardon?
Which type of animal is useful as a model?
Oh, I think I can answer this as a very diplomatic answer. It depends on the question you have, right? I mean that I think the animal, you have, of course, the whole animal with this with all the blood pressure, which you cannot modulate on the iPS cells. You have different questions with mice, which you can use for knockout, which are much easier. You can have pig models, which are more and more uncommon, dog models, which I also worked on in the lab of Stan Natel. So I think they're all useful, but also in this study we saw certain differences in the remodeling. So, for example, we found that whereas in the dog model, L-type calcium current downregulation is based on the protein expression, there you can really see a downregulation of the protein, which we cannot see in the human. So many human studies don't see any L-type calcium channel downregulation, and we also don't see that in these iPS cells. And we see that the downregulation is more due to phosphorylation changes, which we also showed in human cells. So I think there are very specific differences, and it's important to keep them in mind. So, diplomatic answer.
Thank you very much. So with that, sorry. Yeah, go on, Bjorn.
So you're saying that this is a chronic stimulation remodeling. The counter hypothesis would be that your ring's actually just much more mature, as we discussed earlier, because you're pacing more. And so did you do RNA-seq or look at if there's is it a maturation or is it a dedifferentiation?
So that's a very good question, of course. But so that's why we also use the 1 Hertz frequency as a control. That's one point. And the second point is that we can recover the changes we see here. That's also very nice thing in this model, which you can do here. I didn't show the data here, but for example, you see that the L-type calcium current, if you go back to 1 Hertz stimulation, it recovers to normal levels after, again, 24 hours. And also the EHMs, we only measure that for contractility, but they recover after 1 or 2 days. So we can pace them for 7 days, see contractility changes and continue again, and it recovers.
Yeah, but Niels, you know that a fetus beats probably a normal fetus beats at 3 Hertz, not at 1 Hertz.
Yeah, that's true.
And the downregulation of the L-type calcium channel is also part of the maturation progress, because as you shift from a membrane coupling to EC coupling, and the paradoxical shortening is the normal upregulation of IK1. So I'm rephrasing this because we see very similar changes using our T3 plus DEX maturation protocol, but there we also see increase in the mitochondrial function, switching from a glycosylated to a lipid oxidation, and so I just caution to overinterpret the phenomena when in fact they may actually represent something else. Anyway, just a philosophical.
That's true. That's true.
We can discuss
the point for the 3 Hertz is also that was the maximum, and it compare it with also the other in vitro things. But yeah, thank you.
Right. Thank you very much, Niels. One more round of applause, please. Thanks a lot for coming. Great. So with that, I'd like to thank you all for your attention for this session. The session is now closed, and that means it's beer o'clock. So please make your way into the foyer. We have some wonderful beer mugs and some beer for everyone before dinner. Thank you.